Now there are two types of gene therapy. Number one is called in vivo gene therapy, and the other one is called ex vivo gene therapy. In vivo therapy, in vivo gene therapy, what happens? You have a therapeutic gene copy. You insert it into a viral DNA or a liposome in any other form through which it could be transformed to the cell, and then you actually insert it directly into the patient's body. So you see, it says the in vivo gene therapy, the introduction of therapeutic DNA is directly into the patient's body. So the DNA will be inserted into a into the body right away. There will be no cell culture involved. There will be no introduction of the new transgene into these human cells under in vitro conditions. No, you take the therapeutic gene copy in form of a viral or a non-viral vector, and you have injected, you have, I mean, special type of syringes, and then you inject it into the specifically directly into the part, the defective part. For example, if it's a lung tissue that needs a gene repair, you would inject into the lungs. If it's a muscle, then you would go into the muscle, and then inside the body, this DNA will insert, theoretically, it would insert into or incorporate into the specific tissue's genome, and this is how the cells would produce the required or the missing protein. The altered gene would be restored, the functional protein would also be expressed, and the disease symptoms would be diminished. As long as these cells, if it's only one cell that receives this copy and it successfully gets transformed, it will divide into two, and it will keep on dividing. So as long as these cells are keep on dividing, it will, the gene will be there and be making its copies, and the person that is having it will never develop the symptoms. The next step, what happens ex vivo gene therapy? So the difference between in vivo and ex vivo gene therapy is you harvest the cells from the patients. So if we say ex vivo therapy, if we can make convert it into different steps. So again, you have the copies of a therapeutic gene. You already know which particular gene is actually disrupted, it is faulty. You insert the gene into the viral DNA, and then you take the cells. So the target cells are removed from the patients, and you culture it. You culture it in vitro conditions in the lab. Then you transfect these recipient body cells with viral DNA or with microinjection or with microprojectile. You can also use, but we use viral DNA because it's more efficient, and the target cells are now genetically altered, and now it's ensured that these gels, there are, of course, we use markers, either they're frozen markers or they are molecular markers. In this case, mostly it's molecular markers. Once you have ensured that the gene, the transgene is incorporated into the particular genome at a specified DNA region, and it's working, the protein is also working, you place these cells back into this body, into the patient's body, and they would decide the protein, and they would go on dividing, and eventually the disease symptoms will be diminished. Why is ex vivo gene therapy better than in vivo gene therapy? The reason lies here. You know everything in advance. In vivo, you just insert all the things and you hope that things would work, but they might work or they might not work. Time will tell. But here, when you reach this stage, after culturing, after incorporation, after the verification using these marker genes, you know that these number of cells, few number of cells that you are going to transplant into the body of the patients, they have the working functional copies of the gene, and the proteins are also produced. And once they go inside and they start dividing, it will be good for the patient because the faulty gene is already replaced. Will the body produce any immune response against these cells or not? Actually not, because if you see in the step number 2, these cells that we have in cell number 4 are actually exactly the same cells that we harvested from the patients. So the genetic antigenic determinant structure will be recognized as self, and there will be no immune response against it. That's the difference between ex vivo and in vivo gene therapy. And uh, oh, my tea is so hot, sorry. One thing that you should keep in mind is both in vivo and ex vivo gene therapy are in the somatic cell lines. They are not in the germline. Whenever you go for a germline, you would have to go from starting from zygote. So you would have to transfer the gene into a zygote and let it hatch. Okay, vectors, types of vectors for these gene delivery. This is something we have already discussed in detail in genetic engineering. We had also discussed it in production of transgenes, and we have also transgenic animals, and we have also discussed it in plant biotechnology. In case of gene therapy in humans, DNA viruses and RNA viruses are used. So adenoviruses are very much used, and some derivatives of adeno-associated virus, herpes virus, Vaccinia virus. Retroviruses are used, for example, lentiviruses, as I discussed in the transgenic animals production, and some other methods, for example, calcium phosphate transfection, microinjection, electroporation. And these days, everything is getting into nanotechnology, so we are also using ballistic DNA injection, liposomes, neosomes, and naked plasma DNA injections. These are non-viral vectors. The viral vectors, when we use viral vectors, we remove the genes that can cause diseases, of course, but we still have the genes and the makeup which help these viruses to get desired DNA integrated into the genome. Some of the examples of viral vectors are retroviruses and adenoviruses. So they integrate the genetic material, including new gene. There's a retro, they have a DNA, they have an RNA and nucleic acid as a genetic material, and which it can be converting into through a reverse transcriptase enzyme to cDNA, and that cDNA would be then getting integrated into the genome. Adenoviruses are actually DNA viruses, and they are not really, there's no adenovirus that can cause disease in humans. And it does not integrate, the DNA does not integrate into the chromosome. It just gets into the nucleus. There is a very important, very famous story of Ashanti DSilva in 1990, the NIH, United States of America. They performed first clinical human trial of gene therapy when she was 4 years old, and she had a rare genetic disease which is known as combined immunodeficiency, in which you are severely, your immune system is really deficient and weak, and even a small normal bacterial pathogen or fungal pathogen can be potentially strong enough to kill you. So they remove the blood cells, white blood cells from the body, and then, and as we say, if they remove the red blood, white blood cell from the body, this is ex vivo, so it's this type. So Ashanta DSilva undergo under ex vivo gene therapy. They cultured the cells in vitro in the lab conditions, and then they inserted the missing gene, of course, and they performed all the experimental proofs to confirm if the gene is integrated or not. And then what once they were sure that these genes are working and the faulty genes are replaced with a normal workable functional copies, they modified the genetically modified blood cells. They were transferred back into the patient's bloodstream. And it was done in 1990. Was a success story until 2007. And I mean, this is an old, already 13 years, but she's until 2007 she was found in good health and she was attending college, because at this stage she was 4 years of age, and this is after 17 years is, so this was 17 for 21. So at 2007 she was 21 years of age and she was uh, attending college. This is very common in the United States of America. After school you go to these colleges, where professional colleges, you could learn many professions. This proved that gene therapy, as we discussed, is still in infancy, but this proved that gene therapy could be really an answer to many problems that we cannot treat through general medicine. Non-viral approach, as I discussed here, viral vectors, I will also discuss in the next lecture a little more about the clinical grade vectors for gene therapy in humans. But viral vectors, adenoviruses, retroviruses, herpes viruses, and adeno-associated viruses, these are the vectors that are used for gene therapy in humans. But non-viral vectors could also be used. And this A is actually a viral vector, so it says viruses, all the viruses. Then you can use cationic polymers. Cationic means these polymers have positive charge on their surfaces, and since the cell membrane is negatively charged because of the phospholipids, there would be a electrostatic interaction between them, and this way they would stick to the cell membrane, and there are chances that these cationic polymers would get inside not only the cytoplasm but then also into the nucleus, because the nuclear membrane would also be negatively charged. Dendrimers are also kind of polymers, have the same strategy. Then there are some peptides, liposomes, niosomes, gold nanoparticles, a lot of other nano scale formulations could help this. And this the biological uptake and biological activity. You have high nucleic acid loading in these non-viral vectors, but of course there are some disadvantages also associated with it. And this is a multifunctional envelope type nano device, as I discussed. So you could see it's a lipid membrane. So lipid membrane is like a bilipid layer. It's the cell membrane, plasma membrane mimicry into a circle. Inside the circle, these are the transgenes and all the regulatory sequences. And then you have these PEG molecules. As you can see, these PEG molecules, they are given here because this molecule will be taken by the immune system as a non-self and they will attack it. So if you want to provide some masking capabilities to your molecules, you have to give them brushy appearances. So because of the static hindrance of these PEG molecules, the immune system will take really long to capture this guy. And these golden or yellow color arrows, these are monoclonal antibodies perhaps that are used for targeting, because you want this particle to go to the specific cells, not just any cells. And they would recognize specific receptors that will found on those particular cells. Because this is a monoclonal antibody, it would respond to only that particular type of cell receptors. Okay, this was all about gene therapy. Tomorrow in our next lecture, we will be discussing antisense therapy. Antisense therapy could also be included in gene therapy, or not. Both of the types could be as antisense therapy. And then in the next lecture, we will discuss more things, and hopefully by the end of next week, not this week, by the end of next week, we will have completed our course well in time and with ease.